secondary growth activity in the dicot root is completely different with that of the dicot stem. Here it is secondary growth is not seen in the monocotyledonous roots, it is seen in the dicot roots. Here the thickness or the formation of the cambial ring is totally different. Here the parenchyma cells which are formed by the activity of the secondary growth are developed below the group of phloem cells. It will lead to the continuous division and these parenchyma cells which are developed below the group of phloem cells will develop a capacity of continuous division potentiality, and due to which this will lead to the formation of strips of cambium. So for example, below the as we can say, below the phloem you will see the strips of the cambium developed, which for example, if we say the vascular bundle is having 4 strips of xylem and phloem, so we will see the presence of 4 strips of cambium below each 4 strips of cambium below 4 bands of phloem tissue, and which will lead to the which will ultimately undergo division, and which will lead to the formation of ultimately undergo division and go towards the pericycle. Till pericycle they form, and in the pericycle opposite to the primary near the pericycle, which is opposite to the primary xylem, which will undergo a differentiation into the cambium. This cambial tissue undergo periclinal division. The periclinal division of the periclinal division of the cambium is occurring, and here this may lead to the formation of secondary xylem towards the inner side and secondary phloem towards the outer side. Inner side and secondary phloem towards outer side. As a result, there is a formation of undulated. As a result, there will be a formation of undulated ring of cambial ring, which is occurring towards the center of both the xylem and phloem occurring. So what are the elements which are present? Here also you will see the presence of peridum, which is present towards the outer side. Peridum towards the outer side, and you will also see the presence of cork cambium is present, and this cork cambium is present towards the peripheral side of the pericycle. It is present towards the peripheral side of the pericycle. Peripheral side of pericycle. As a result of the activity of the cork, it is due to the activity of these cells in the due to the activity in the cork cambium tissue, the cork tissues are present to be occurring in the outer side of the tissue, and you will see the presence or existence of the occurrence of secondary cortex towards the inner side. Due to the pressure created by the continuous continuous secondary growth, because of the pressure created by the inner secondary vascular tissue, what will happen? There is a lot of pressure existing on the internal tissues like cortex, endodermis, with all that's what will happen. Because of excess pressure they will break down, and what we said earlier in the dicot stem, because of the breakdown of the epidermis, there is an exposure of cortex or internal tissue to the outer atmosphere. So to avoid that internal tissue, we will develop a structure called as peridum, and in some cases, as the cork tissue is a dead tissue, they are arranged closely, we do not have the chances of gases exchange occurring. So at this time you will see the presence of lenticell functioning here. These lenticels help in the exchange of gaseous material or food material, and these lenticels at the time of dicot root secondary growth, these will occur in the form of pairs, and in these lenticels are later on, as the multiplication goes on, develop into a transverse bands these are developed transversely in the form of bands, and it is on the rough areas on either side of the lateral roots. Now this lenticels, which helps in the exchange of gaseous material, this may be occurred transversely in the transversely extended in the form of pairs, and roughly towards the in the rough areas of the lateral roots, and ultimately help in the occurrence of the secondary growth. So what is there in the dicot root secondary growth comparing to the dicot stem is, in dicot root the formation of the parenchyma cells occurs below the group of phloem cells. If there are 4 bands of xylem bundles, then there will be 4 bands of cambium tissue present below the 4 phloem bundles. I mean, if there are 4 xylem and phloem bands, then there will be 4 cambium rings present, and this will undergo continuous periclinal division, and these divisions of periclinal will move towards the pericycle. This pericycle will undergo divisions, and this will lead to the formation of cambium, and this periclinal divisions of cambium will lead to the formation of secondary xylem towards the inner side and secondary phloem towards the outer side. Now this because of the secondary growth, as we said in dicot stem, here also a lot of pressure is existed on the epidermal tissue. So because of which the breakage of the epidermal tissue occurs, and in order to protect the internal cortex tissue or internal tissues, a layer of protective covering is developed, which is more or less similar to epidermis, that is called as periderm, and towards the inner side you will develop a cork cambium. This cork cambium tissue is present towards the peripheral areas of the pericycle. That if there is a continuous division, periclinal division or meristematic activity is going on, then the cork tissue is developed towards the outer area, and secondary cortex is developed towards the inner side of the dicot root. Now because of this continuous differentiation, there are possibilities of cork tissue is there. So cork tissue, as we said, it is a dead cell, closely arranged cells without intercellular spaces. So there are possibility of exchange of gaseous material is not possible. So that is taken up by us. Cells, those are called as lenticels. What we said, these are sand shaped spot like structures. These occur normally in dicot root in the form of pairs. They will come, and these pairs are occurring transversely. These are occurring transversely and extend in the rough areas of the lateral roots and undergo the function of gaseous exchange, which stomata can do. So this is about the secondary growth in the dicot root. So here we end with that of the dicot root and dicot stem secondary growth.